Good evening, my dear students. Today we are discussing about one of the topic in genetics. So hereditary hemochromatosis or genetic hemochromatosis. So it is also called as a HHC, hereditary hemochromatosis. Hereditary hemochromatosis is one of the topic in our genetics, HHC. So first of all, this one is hemochromatosis related to the blood, especially related to the iron related particles. So now we'll discuss about how to develop the hemochromatosis, hereditary hemochromatosis, how to transfer one person to another person, means one generation to another generation. So how many members are carriers? So which one is completely affected from this one? Who is free from this type of problems? We'll discuss about every character. Before we discussing about that, we will go with the introduction part. In introduction part, so hereditary hemochromatosis is inherited predisposition. Mention one word. Pre-inherited is a predisposition, absorption, excess iron from the right. From the small intestine, willies are absorb the excess amount of the iron. What is the normal body iron? Just 3 to 4 grams. But due to this hemochromatosis process, so approximately 20 to 60 grams of iron is reached to our body by the absorption from our body. How to reaches? Due to the one gene. What is that gene? HFE gene. Due to the HFE gene. Due to this HFE gene, the HFE gene, it is occurred in our body at the chromosome number 6. At the chromosome number 6. At the chromosome number 6 is a HFE. HFE. So the HFE gene is located at the 6th number, and the HFE gene, it is the occurred in the mutation. So it is the increases the iron content due to the mutation. So mutations in the HFE gene, the most common case adults onset iron is overload, increases the iron concentration in our body due to the HFE gene. So due to this one, so not HFE gene is iron increase, not in the blood, sometimes stores in the liver, pancreas, heart, joints, and another body parts also. It leads to the cell injury. Sometimes cell death also occurred in our body, right? Cell damages, cell injury. So this one is occurred in the body. So what are their prevalences and the incidence? So first of all, you just check this. It is the autosomal recessive inheritance, not allosomes. Actually, in the human body, 23 pairs of chromosomes is present. Out of 23, 22 is the autosomes and 1 is the allosome. So here this autosomal related disease, that is a autosomal related disease, and HFE associated with the hemochromatosis. So more than 90 cases. So here at which point sometimes is a single gene only is affected. So sixth pair means two chromosomes is present, two chromosomes. So here sometimes single gene is affected, sometimes multiple genes also affected. So which gene is affected? So genes, different types of genes are affected in our body. We'll discuss about every characters of the genes. So first of all, just check this point. In this point, mention one word. So codes are mentioned related to the genes. Single gene mutation occurred in the part of the C2A2Y, C2A2Y and H63D. H63D, these two genes are going to mutation. These genes are located in the sixth position, HFE gene. These two genes are going to mutation in our body. It leads to the hemochromatosis, hereditary hemochromatosis process. So what is the C2A2Y? C2A2Y is related to the cysteine is replaced with the tyrosine. Cysteine to tyrosine is substituted. So cysteine replaced with the tyrosine and histidine replaced with the aspartic acid. These two codes are developed in the body. Just protein containing amino acids are change their position. Replacement of cysteine replaced with the tyrosine, histidine replaced with the aspartic acid. These type of problems is develops, and it leads to the increases the iron content in the body. It leads to the blood or genetic hemochromatosis occurred in the body. Now we'll discuss about different character. Just check this point. So here first one is a blue color. Here only one part, just check this part only. So blue color. So in here blue color it indicates the normal persons, and brown color it indicates the carrier persons. In case mother and father both are suffered from this type of problem, just imagine. So here mother and father both are suffered from this type of problems. So in case father only one gene, father only one gene XY, and mother here XX, two genes. So here for example in father Y chromosome, in mother X chromosome. So first of all what happened here, XX dash occur here, and the second one is XX, not Y ma, XX. So here one is the carrier, one is the free from the disease. Just check this one more point related to the Y and X, X dash, Y dash, and here Y dash and X. So who suffer from the both genes? They are the affected persons. And here one person is unaffected person, and one gene is presence in the body, and here also one gene presence in the body related to the inheritance problem. They are the carriers. Simply say like that, simply just like that, just check once this process. So here first one is the blue color. What I'm saying, already mentioned one point, blue color it indicates the free from the disease. Here in the given structure, this boy is free from the disease, and this girl is affected child, and remaining two is the they are the carriers. Sometimes the type of characters is exposed, sometimes is absence also, carry forward into the next generation also, like so this is about the autosomal receive inheritance process. And this, what about the HFE gene? Already mentioned this point. So HFE gene is on the sixth chromosome. So this one is the involves in the iron hemostasis, inheriting, transferring receptors. So the basolateral membrane intestinal epithelial cells. So this one is increases the protein. The HFE protein normally limits amount iron uptake from the gut regularly. Amount iron stores in the tissues in normal condition. This one HFE is going to mutation, defects is occur. So excess amount of iron is absorbed from the gut and small intestine, stores in the different parts of the body. This is about the HFE gene. So in the HFE gene, just observe this structure. So the HFE, how to going to mutation. So first of all, liver. So from the liver, this is the normal condition. This is in the normal condition. Just imagine. Wait. Right, these two parts just observe. So in normal condition the cells are distributed to like this, and lumen cells are reaches to here, but here abnormal, the cells are not reaches to this area. So the macrophages releases which cells, that cells are reaches to this area, right? So this is the mutation, way of mutation. So mutation means sudden changes occurs in the genes due to the any reasons. Lot of reasons are there. We'll discuss about different types of reasons. So what are the reasons? Sometimes this type of characters is developed, depends upon the age, depends upon the sex, male or female, iron, dietary iron, excess iron intake, alcohol consumption at the time of the menstruation and pregnancy, unknown factors also, alcohol, sometimes hepatitis, microorganisms, and diabetes characters, and different types of characters are to develop this type of hemochromatosis process in the human body. So due to that, what are their clinical manifestations? Means simply signs and symptoms. First one is the weakness. Why it related to the blood? And the next one is skin discolorization, gray color, abdominal pain, joint pains. Why? These are the stores in the joints also. And diabetes and different types of problems is occurred in the body. At least you should mention the 5 or 6 points mentioned in exam point of view, enough. Okay, these are the clinical manifestation. So just check this. So normal hand color is this is the normal color. Due to the excess iron, turn into the reddish color. Turn into the reddish color. This is the one of the example for you. So how to estimation of this type of characters. So first one is the identified diagnosis by the, so first one identified by the ferrous accumulation, means iron accumulation. And the next one serum ferritoin. And the next one menopausal woman, post or pre. And the next one normal values of the iron levels. And the next one estimated by the CT scan technique, liver biopsy test. And the next one simply and DNA C2A2Y and H63D gene mutation techniques also used to the determination of person is suffered from the problem or not is detected by the following method. So treatment is first one is a venesection treatment. Venesection treatment means withdraw of the blood, draw the blood, venesection. And the next one DFO method, and DFO defor-examine method. And avoid the iron supplements, red meat, and avoid the alcohol, tobacco. Here very important method is venesection method. What is the venesection method? So in venesection method means simply removal of 500 ml of blood, removes the 250 mg of iron, and do weekly unit iron depletion. And the hemoglobin percentage, ferrous percentage, just check the normal values. So this process is done up to how many days? So up to every 3 months. So this is the venesection process. The venesection process simply removal of blood and iron content from the body. This is about our topic, and tomorrow we'll discuss about one more topic. Thank you.